Welcome to today's webinar. Today I'm going to talk about the construction of custom repeat library for genome annotation. I will not go through each detailed step because my main intention is to explain the background and rationale of our method. In this case, you would understand when you are doing your work, you know which step is critical, which one is not. So this is today's outline. I will first talk about the classification of transposable elements. So in a genome, there are structural components like centromere and telomere, which is important for the cell division or the integrity of the chromosome. The other components are genes. Usually we refer this to protein coding genes, but there are also non-coding RNA genes. It is very often the gene space in the genome is much smaller than the intergenic sequence, except a few, very few species with very small genomes. So according to the sequence, whether they are repetitive or not, we could divide the genomic sequence into a unique sequence, which include the genes and the regular regions. But it is very often that the repetitive sequence in the genome occupy much larger space than unique sequence. So this repetitive sequence, including gene families, tandem repeats, such as centromeric repeats, telomeric repeats, and also transposable elements. So in most eukaryotic genomes, there are large amount of transposable elements, also called jumping genes, because they not only sit in the genome, they can change their position, and when they're jumping from one place to another, it often increases their copy numbers. So the transposable elements were first discovered by Barbara Minhag. She won the Nobel Prize for this discovery in 1983. So in 1940s, she used the maze as a model system. So as you can see on this slide, if the anthocyanin gene is functioning, the clonal is purple. And if a transposon inserted into the relevant gene, the clonal lost the anthocyanin, so it appears to be yellow. However, when the transposon jumps out of the gene and the gene function recovers, so you see this all purple spots because the pigmentation appears again. According to the transposition mode or the intermediate of transposition, transposable elements can be classified as two classes. So the class 1 is called return transposon, also called RNA transposon, because they use RNA as an intermediate for transposition. The class 1 elements can be further divided into long-terminal repeat element and a non-LTR, so the long-terminal repeater is called LTR, and so the other cluster is non-LTR elements. The LTR elements can be further divided into copy-like and GPs-like based on their gene order. And also the third class is the endogenous retrovirus, which contains the enveloped protein. That means they could become a virus. I want to spend some time on the long-terminal repeat element because they are the largest component of plant genomes. So a LTR element contains the long terminal repeats in the same direction. And also, when they inserted in the genome, they duplicate a small piece of flanking sequence, which we call the target site duplication, or TSD. So the internal region of a return transposon, LTR return transposon, include proteins that are important for the reverse transcription and the integration of the element. So when the AOTR element started to express, they created, transcripts. Then the transcripts are reverse transcribed into cDNA, and then the cDNA inserted into different places in the genome. So as you can see, when a new insertion formed, the old element is still there. So this is called a copy and a paste mechanism. The class 2 elements also called DNA transposons because they use DNA directly as the intermediate for transposition. And it can be divided into two subclasses. The subclass 1 uses cutting and paste mechanism. So cut the element out, then insert the element into another place. And the class 2, which is just discovered like less than 20 years ago, is use a replicated mechanism. They don't cut the element out. They use a for a mechanism that integrated two sequence pointed together and replicate that element. I'm not going through the details. So a typical class 2 element has a terminal inverted repeat, and they encoded transposase. The transposase were binding to the terminal inverted repeat to cut the element out. And also, like the AOTR return transposon, they created a taxa duplication upon the insertion of the elements. Of course, there are some exceptions. For example, the helitron I mentioned previously, they don't create a taxa duplication. So here is an example of the hierarchy of transposon classification. So for example, I said a mutator-like element, so it belongs to class 2 or DNA transposons. It also belongs to the subclass cut and paste. Then it belongs to the order of TL element, which means it has terminal inverted repeat. Next is the subfamily, so it belongs to mutator-like subfamily, also called MULE. And the family name is mutator. So the mutator family contains more than 10 subfamilies. We call the MULE2, MULE1, MULE2, MULE3, until MULE11, MULE13. And each subfamily contains many individual elements, which is there is insertion in the genome. We call this individual element. Having said that, I have to point out that the definition of family or subfamily is somehow arbitrary, because different people could have different standards for family or subfamily. So, for example, Tom Swiggle and his team proposed that if two elements show 80% of identity in 80% of the element sequence, these two elements belong to the same family. So this is well accepted, but so as I told you, so you could define in your way, which is convenient or makes sense. So DNA transposon, this is how they are transposed. So first, they will make RNA and then make transposase protein. The transposase protein will bind to the ends and cut the element out, and then insert it into other place in the genome. So this is different from return transposons. According to whether, because not all the transposons, they include transposase, some of them not. So if they encode transposase and can move by themselves, we call it autonomous element. And otherwise, we call it non-autonomous element because they rely on the transposase from the autonomous element to help them to move. However, non-autonomous element, sometimes they also encode some protein, but it's not a transposase. When the two elements, the one element just jump to another, form a new copy, so these two new copies are identical. After they're sitting there for millions of years, the mutation will accumulate. And then you can see these two elements become less similar. So that's why I say the definition of family or subfamily is very arbitrary. Because as you can see, when the two elements just formed, of course we will define them as the same family or even the same subfamily. But after several million years, they don't look so similar. Then according to the sequence similarity, we may define them to different families, but they are indeed derived from a single element. So when the elements stay in the genome even longer, more than several million years, they started to have more mutation or more insertion or deletion. So in this case, the intact element structure won't be remains. So for example, this element will become a truncated copy. So the other element will become a fragment. So in this case, unless this fragment encodes transposes, or there is an intact copy in the genome for us to compare with, otherwise people have no way to recognize whether this fragment belongs to a transposon or not. So that's why I say, the transposons are only recognizable for a few million years. After that, they may still physically be there, but you just cannot detect them. So next, I will talk about the abundance and the insertion preference of transposable elements. As I mentioned previously, transposable elements are the major component of many species. Of course, there are some exceptions. For example, the Arabidopsis. Arabidopsis has a very streamlined genome. It's only a little bit over 100 megabases. So about 10% of the Arabidopsis genome are transposable elements. But if we look at rice, which is 400 megabases, half of the genome, above half of the genome are transposable elements. And human, ourselves, contain the half of the genome are transposable elements. So the small transposon like allele has 1 million copies in everybody's genome. So if we look at maize, which has a genome size similar to human, 85% of the genome are transposable elements. And among them, 75 of the genome size are ALTR regional transposons. So in plants, the amount of ALTR regional transposon largely explains the variation of genome size. So in a genome, if it has a reasonable size, usually there are thousands or more families of transposable elements. But their abundance, they have different copy numbers and different size. So here is the example from rice. We detected a total of over 2,000 families of elements. And if we look at the contribution of them to the total repeat size, you can see here. So the top 10 elements contributed to almost 20% of total repeat size. So only 10 elements, 10 element families. So if you look at it here, the top 100 elements contributed to over half of the total repeat size. And if you look at the least abundant 1,000 families, it only contributed to 6% of the total repeat size. So this tells us if you have time limitation, you don't want to work on all the repeat size, you can just focus on the most abundant ones. Then you can get the majority of individual copy of them. On the other hand, the low copy number elements are also important in a certain sense. For example, in plants, probably so far there are like less than 100 active elements discovered. And most of them are very low copy number elements. This is because when a transposon amplifies to high copy numbers, so the genome will sense the threat of the transposon and sense the danger. So it activated the silencing mechanism. So most high copy number elements are no longer active. As a result, it depends on your research purpose, whether you work on the highly abundant elements or you work on the low abundant elements. In the genome, most of the transposable elements, they have something called a target specificity, which means they are not inserted in a random pattern. So most of them has a selection of insertion preference. So here again is the rice chromosome. This is rice chromosome 1. So this is the physical distance from the end of the chromosome. Here, the red arrow indicated the position of centromere. So as you can see here, the red ones here, red line, indicate the abundance of class 1 transposon. So as you can see, most of the class 1 transposon is concentrated in the region, pericentric region, and form Horticulture. And in contrast, the green line is the class 2 DNA transposon. So most of them has low abundance in the pericentric region, but more located on the chromosome arms. So in general, the DNA transposons are frequently associated with genes, and the region transposons are more likely located in the heterochromatic regions. But this is not absolutely true. There are always exceptions. So here is the example, a DNA transposon called DS inserted into an intron of the gene related to lignin synthesis, and of course, the frame shift of transcript then eventually lead to the reduced amount of lignin in the plant. So DNA transposon or some of the region transposon could be inserted very close to the gene or inside the gene. On the other hand, some other elements, they don't like inserted in the gene. They inserted into themselves. So this is a strategy for them to survive, because if they inserted into other transposons, they don't create a harmful effect to the host organism. So this is a safe haven for them. But because of the nested insertions, it creates a problem for the detection of the elements. So next, I will talk about the relationship between TEs and genes. We talk about that they could be inserted, the TEs could be inserted beside the genes, but they could also maybe serve as genes. So here is the example of Arabidopsis plant, and there are two genes called FHY3 and FAR1. So these two genes, when you knock it out, it lost the response to the far red light. So for example, a plant in the dark should have long cotyledon because they want to get out to see the light. So when they see the far red light, they are not supposed to elongate it. They should be short. This is the white type. And this is, if you knock out the FHY3 genes, so the plants don't see the far red light and they still keep elongating. So this indicated that this gene is a functional gene. So later on, when people cloned this gene, they found that these two genes are actually closely related to transposase. So they become a real cellular gene instead of a transposon. So this is how it works. So as I mentioned before, the transposase of DNA transposon, they would bind to the end of the element. So in this sense, all the transposes are DNA binding proteins. So they can easily evolve in the transcription factor, which are also DNA binding proteins. So an element inserted in the genome, but then they gradually lost the structural feature, for example, the terminal inverted repeat of transposon, and then the target site duplication also becomes degraded. But because they have a cellular function, the transposase domain, the DNA binding activity is remained, and they become a real gene. So not all the transposase-related protein in the genome are transposons. Some of them, we call it domesticated transposon, and this process is called domestication of transposons. In addition to contribute their transposase for cellular functions, some transposons, they could duplicate the real gene, the other cellular genes. For example, here is a PAC-MULE element which belongs to the mutator family. It duplicates a sequence from 2 different genes from rice and combines them together and form the new open reading frame. So when the transposable elements duplicate other cellular genes, they have the potential to create a new gene. Next, I will talk about how do we detect transposable elements. So if you have a new genome, the best way is you should remove the transposable element first before the gene annotation. For example, as I told you previously, the maize genome is over 2 gigabases, and 85% of the genome are transposable elements. So if you can mask this sequence out before you do gene annotation, you only need to look at 15% of the genome, which reduces the use of computation power a lot, and also save your time. In addition, because sometimes transposable elements are inserted close or even inside the genes, and some of them has coding capacity. So if you remove them, you could minimize the opportunity of misannotation. For example, you put one of the transposon exon fused to the gene exon to form a fake exon. Of course, sometimes the transposable elements serve as exon of genes, but this chance is not very high. So the best way, if you have a new genome, is to construct a repeat library for this genome and mask the transposon out. So maybe you will say, I don't want to bother to construct a custom repeat library. Can I just download transposon elements from RepeatModeler, then use this sequence to mask my genome? Yes, you can. So if you have a closely related species, for example, if you have maize repeat library, and then you are sequencing the wild species like like TES, then you can pretty much use the maize repeat library to mask TES. But otherwise, if your genome is more than a few million years diverges from the new genome, from the genome you have a repeat library, you should build a repeat library for your genome. This is because I mentioned before. So after a few million years, the transposable elements become not recognizable. So the new transposons in the new genome may not exactly the same as that in the older genome because the transposable elements has low constraint, so their sequence evolve much faster than the gene sequence. So they are not conserved except in the closely related species. In addition, if you download a repeat library from rabbits, that library is very, very large. Usually it's like 20 megabases or even larger. And if you use that to mask the genome, it will take a long time. So a custom repeat library usually are much smaller in size. So to collect repeats, of course, it's like any other search. You want both high sensitivity and high specificity. But here, we consider the specificity is more important than sensitivity because you don't want to mask a transposon that is not a transposon. You mask something important, either a protein coding gene or nucleotide out, then you lost this gene. So in this case, a over annotation of genes is probably better than under annotation. So to detect the transposable elements, there are two methods. So one is the homology-based. So for example, the repeated mask is based on homology of the unknown sequence to a known transposable element. So if you give repeated mask library, it will mask the repetitive sequence out. So the other method is called the de novo method, means it does not rely on homology to non-transposable elements. It is just based on homology of the unknown sequence themselves. So if they have homologous sequence in the genome and they appear to be repetitive, it's considered to be a repetitive sequence. The second method is called structure-based. So that means using the non-structure feature of transposable elements, for example, the terminal sequence. So this method has been developed for like mice, miniature inverted repeated transposable elements, ALTR elements, signs, and hedgehogs. So the de novo method based on homology is very powerful because, as I showed you before, the most repetitive transposable elements contributed to a larger part of the plant genome size. If you get the most abundant ones, you get a larger part of transposon in the genome. So for the de novo method, any repetitive sequence will be recovered. This is good or bad. The good thing is you get all of them. The bad thing is it will also collect like something like gene families, but we don't really want gene families to be masked out before gene annotation. The other disadvantage of the de novo method is that it cannot identify low copy number transposable elements because it's totally based on copy number. And also, most of the de novo method does not classify elements. So the only thing you know, it's repetitive, and know it's likely transposon, but you don't know what it is. So this increases the uncertainty of the element you collected. In contrast to the homology-based method, the structure-based method does not rely on copy number. So it relies on the structure feature such as terminal repeat, terminal sequence. For example, the ALTR starts with TG and with CA. Also the taxa duplication, because each subfamily of transposable elements, they have different lengths of taxa duplications. So this method, it can identify both high copy number elements and low copy number elements because it does not rely on the copy number. But so when an element becomes old or degenerated, the structural features does not remain intact, then the element cannot be detected. Also, the structural-based method, it depends on the knowledge of existing elements. For example, if I tell the program to detect the AOTR return transposon, it will detect the AOTR return transposon. So it will not detect any novel elements that are not discovered previously. So these are the limitations of structural-based methods. Okay, so finally, we will talk about the construction of repeat libraries. So several years ago, we developed a protocol to construct the repeat library, and here is the website. And because I'm a plant person, so this protocol is optimized for plant genomes, but you can also use for other genomes. So our plan is we'll have another update later this year. So in this protocol, the first step, we are trying to collect mice, which are miniature inverted repeated transposable elements. This is not a taxonomy term. This is including a group of DNA transposable elements, which of course, because they are small, they are non-autonomous elements. And a feature of mice is they are frequently associated with genes in the flanking sequence or inside the genes. So we put this step first because if you need to identify the small element first to minimize the chance of a misclassification of elements. So for example, as showing here, if a mice, the small element, inserted in the bigger element like an AOTR element. So if we don't identify this element first, if we identify this big AOTR element, so this entire sequence would be considered a AOTR return transposon. So later on, if you use this entire element to mask the genome, and then what you can see when it matches to this sequence, it appears to be this sequence is a piece or is a fragment of an AOTR return transposon, but in fact, it is not. So when you identify small elements first, so next in this situation, you will know it's a small element inserted into a big element. It's not a piece of fragment of the big element. So to collect mice, we used a method called Mite Hunter, which was developed by Zheng Han and Suesela. So we used this method because the positive rate of this method is low, and the computational requirement is reasonable. But of course, for a big genome, it will take a long time. So I suggested that for large genome, more than 500 megabases, you should use half or even 1/4 or 1/10 of the genome to run it. Because usually, mice have quite a high copy number. So if you use partial genomic sequence, it will not reduce the sensitivity a lot. So the next one, as I told you before, the AOTR transposons, they are the largest component of plant genomes. And most of the AOTR transposons, they are very large in size. That's why they could be the major component of plant genomes. But there are a group of AOTR region transposon called the terminal repeat region transposon in miniature, they are quite small. They are kind of act as a mice which they frequently inserted around the genes. So there are many programs developed for identify AOTR region transposon, and most of them, they have very high sensitivity. But a common problem of them is they have high false positives, which is very problematic. So as I'm showing here, so the first step of detecting AOTR elements is to detect two similar sequences, which is close by. So as I'm showing you, so if two DNA transposon inserted flanking a gene, so these two DNA transposon could be considered as a pair of AOTR of a AOTR sequence, and then the genes that flanked by this DNA transposon could be considered as the internal region of the AOTR elements. And then if you use this entire structure to mask a genome, you will mask this gene out. And if this gene belongs to a gene family, then this will be horrible because you are masking all the gene family members out, and you will lose a lot of genes. So for this reason, we recently developed a package called AOTR Retriever. It's just published in Plant Physiology. So in this package, we used the output from several, AOTR identification programs. So in this case, we can maximize the sensitivity. So then we carefully look at the possibility of false positives, the different types of false positives, and we developed a filter to filter them out. So in this case, the specificity is very high, usually higher than 95%. Meanwhile, we also developed a code to identify the elements with non-canonical terminal sequence, because most of the AOTR elements are starting with TG and with CA. But we found that there are elements with different ends than TGCA, and most times these elements are actually inserted around the genes, so it's very important to gene annotation. So we developed methods to identify this kind of element too. So the output of the AOTR retrieval will provide a GFF for all the intact elements, so it's very convenient for gene annotation. And it also provides the estimation of the time of the insertion of the element. And so because the region transposon has larger size, and if you collect all the region transposon in genome, the size of the library is very big. So we also are providing methods to build non-redundant library, reduce the size of library. And this package could also use for the correct long reads like a pack-by read. So you don't have to have a genome assembly before you use it. It's multi-sorting, so you could split your genome into small pieces to run the job, so which solve the problem for larger genomes. So after we collected the two most abundant classes of transposon in plant genome, one is the mice, another is the AOTR region transposon. So both of these methods are structure-based. So then we use a homology-based method to collect the remainder of repeats. So we use the RepeatModeler. RepeatModeler is a single sorting program, so you cannot split your genome. This is the drawback of the program. But the good thing is it automatically classifying repeats, that is the code classify. Of course, not all the classifications are correct, but the majority are fine. So this report module, of course, itself is not a de novo method. It uses the output from two de novo program called a recon and a repeat scoot. They use a different method to identify repetitive sequence. So yeah, because the repeater modulo is relatively slow, so we recommended you run it after you collect the mice and AOTR from mice handle and AOTR sequence from AOTR retrieval. So then you use the mice and AOTR to mask your genome. So after mask these two classes of elements, your unmasked genome size will become much smaller. So in this case, so it's possible to run repeater model. Even if so, in this case, so for the larger genomes, we recommended you proceed stepwisely. That means for a larger genome, you use a small portion of the genomic sequence to run repeater model first. And in this case, you get the most repetitive sequence. So then you use this. So then you use the output from this small portion of genomic sequence to mask the rest of the genome to reduce the unmasked size of the genome, and then use it to run repeater model again. So if the genome is really big, you can separate it into multiple steps to make the repeater modulo run in a reasonable time. Okay, as I mentioned at the beginning part of my talk, so even if some real transposon like PAC-MULE, they could contain gene sequences. So in this case, you definitely don't want the gene sequences to be masked out from the genome. So this is very critical for you after you collected a repetitive sequence. You remove the possible gene sequence from repeater libraries. So for this reason, we build a package called put excluder. So you blast your repetitive sequence against the plant protein database. Of course, you could blast against any protein database you like. For example, if you are working on human, you use human protein database. So then you feed the blast output to the put exclude program. Then it will remove the gene sequence. So the default, because if you have a gene fragment, it's likely the closely related sequence is also gene sequences. So the default is they remove the matched part, and also it will remove the 50 BP flanking sequence. But you could, depending on your purpose, you could set the how long you will remove the sequence. Because if you run, say, I don't want really miss any gene sequences, you can set the flanking sequence longer to make sure no gene sequence are masked out. So after you remove the gene sequences from your repeater library, so now you see you have the mice sequences, you have AOTR sequences, you have the classified transposon sequence from repeater modulo. These are most likely the real transposon sequence. usually there will be considerable portion of the repeats from RepeatModeler which are not classified. These are mostly the represented the ancient transposable elements or the transposable fragment of transposable elements. But of course, we cannot rule out the possibility that it contains non-transposon sequence or even some novel gene families. So for this portion, you should really use it carefully. So if you say, I don't really want miss any potential genes, so you don't use it. But if you say, I want my gene annotation really high confidence, so you could use it because the possibilities, most of them are real transposable elements. So it depends on your purpose and your requirement. Okay, so this is end of my talk. I want to thank National Science Foundation and the Michigan State University provide funding for our repeats identification project. I also want to thank all the users who use this the protocol. I especially want to thank these people. There are too many of them. I don't want to name each of them. They provide the feedback of their experience and help us to improve the protocol. Thank you very much.
Thank you. Fabulous. Thank you so much for that wonderful talk. We've got a couple questions here already received, and certainly if other people want to unmute and ask, let me know. I'm not sure if you would know the answer to this, but has anyone published a repeat library for Mesidae, which would be the ant group? Where would be the best place to find out where to find those sort of repeat libraries?
Well, there are, so the easiest way, I think everybody knows RepeatModeler. So you should look at RepeatModeler first. I'm not aware, but to be honest, I'm not an animal person, so I'm not familiar with the stuff in the animal world very well. I'm not aware of this library.
Okay. Another question I received, are the transposable element divergence rates stable across eukaryotes? Arthropods have a shorter generation time, so would that cause faster divergence?
The short answer is their divergence is not the constant. So in different lineage, they diverge in different speed. Even different transposons, they have different speed. Even the different region of the same transposon, they also have different speed. This is because, say, the transposon have coding region like a transposase region or have non-coding region. So usually, the transposase will diverge slower than the non-transposase region. And even transposase, the most critical part, like the catalytic domain, diverges much slower than the other region. So it all depends.
Okay, fabulous. You mentioned that given the distribution of elements and insertion preferences, would you agree that DNA transposons are probably the most important to focus on for gene annotation, or would you recommend that everyone really try to mask all types of families?
Masking there more than the interference is also to reduce your, if you mask it, you don't have to worry about that. Yeah, of course, the DNA transposon is more important in terms of gene annotation because they are close to genes. Among the regional transposon, like sign or copier like LTR element, they are also sometimes inserted beside the genes, so they are also important.
Okay, great. Are you aware of anyone that has used repeats to resolve phylogenetic relationships instead of using protein-coded genes or ultra-conserved elements?
Usually, people don't use, they could use the repeats, but it's not really use their sequence. It's really used because, say, especially return transposon, after they insert it, they will never excise. They stay there forever until there are too many mutations accumulated, then they are not recognizable. So in this sense, so the closely related species, they share a lot of insertions. So the distantly related species, they share less. So then you could use the conservation of the insertion pattern to infer the phylogeny.
Neat, okay. Have you been able to estimate the amount of genetic diversity that's being driven by transposable elements as opposed to other mechanisms like mutation and recombination?
I think I didn't compare them side by side, but I would say probably transposon is because it depends the activity of the transposon. So if a transposon is active in a lineage, then it really creates a lot of mutation. But if a transposon is largely silenced, then of course it cannot compare to other mechanisms. So it all depends. In some of transposon I worked previously is called MPIN. It's very active in rice, and in one of the cultivar, it created like just 1,000 insertion in just like a few hundred years. So in this case, you can see this is really powerful, source of mutation for genome evolution. Yeah, but just as transposon themselves, so the portion of the mutation created transposon is so variable along different species or different lineages.
Great. We've got a few chats going here with some people sending some chats out about a previous ANT repeat library that is available. We've also got some information about genomic repeat abundances that contain phylogenetic signal. So that might be helpful for those interested in those questions. I also have another question here. There might have been a little bit of misunderstanding about towards the end. Why was DNA transposons not a part of the library creation and masking, or was that part of your pipeline?
It is. So the mites, we call it mites, but it's all the mites are actually non-autonomous DNA transposon. So the DNA transposon is different from the return transposon. So DNA transposon is like the autonomous element. The copy number is much lower than the non-autonomous element. So the majority DNA transposon in the genome are the non-autonomous form, which we call mites. So mites including the majority of non-autonomous DNA transposon.
Okay. And I know we're getting to the bottom of the hour here. I think I've got a couple more questions coming in. How much does having a draft genome assembly affect repeat annotation, and is there a minimum level of contiguity that's recommended? I think you spoke to this a little bit in terms of the fact that you could just do this pipeline on PacFile long reads, but maybe you have some other thoughts there.
Yeah, so basically, if you want to recognize a transposon, so your reads should be longer than the transposon. So I would say the majority of transposon is smaller than the length of PacBio reads. So PacBio reads, as long as you corrected it, it's totally fine for identifying any transposon.
Okay. And I think we might make this our final question unless anybody wants to send something else in. Is the prote excluder package that you spoke about, is it sensitive to transitive error in the fact that if the protein database was generated from another organism where they didn't properly mask for repetitive elements first, would that cause you to be less likely to properly mask your genome?
So here is the concern is the protein database contain a transposon, right?
Yes, yes.
So then you mask the real transposon out. Yeah, first of all, I don't think this is really would be very toxic because you just missed the one transposon. Second, so the protein library I provided, I kind of curated it. So the chance for transposase miss there is low.
Okay, fabulous. Well, I think this brings us to the top of the hour, so I just want to go ahead and thank you for this wonderful talk and all of the discussion that was able to be generated. And I want to also remind everyone next month's webinar will be by Carson Holt, who will talk about maker and gene annotation. So this was really the perfect talk to have right before that because it's a critical step in good gene annotation. So I look forward to having everybody on next month's webinar, and thank you again, Dr. Jiang, for your presentation.
Thank you for organizing it. So Anna, we're good?
Yes, sorry about that. Yes, I thank you again. This was really great. I've actually learned a lot. This has answered a lot of questions I've had all along. So so thank you for your help with this and all the help you've done for the community generating these workflows. So I thank you, and certainly I may have to be in touch with additional questions of my own, but I think this turned out really well. So thanks for all the work you put into it.
Oh, you're welcome.
Yes,
I'm glad there are many people attend. I thought that maybe like there's 3 people.
No, we tend to have our numbers vary depending on the month, but we also have a large crew of people that watch the recordings pretty much solely because of the time. This is either during a bad time of the day for them, they're in class or something like that. So we tend to get a lot of YouTube visitors. So I was really pleased that you were able to let people record this as well and listen to it that way. So,
okay.
So yeah.
And I found a typo in my slides. Can I send you the new one?
Oh, yes, definitely. That would be no problem. We're happy to post the new slide deck. And then I'll make sure to get you this, the recording in case you guys want to post it with your future workflows, because sometimes I think it helps people to have some of this background, and maybe that would be something that you want to distribute to people before you're able to get those new protocols up.
And I'll certainly we'll make sure that you get the link to the YouTube video once I get it posted up also.
Yeah. All right.
Okay, great. Thank you.
Yeah, and I hope you have a wonderful day, and thanks for starting it so well with us.
No problem.
All right, take care. Bye-bye.
Thank you. Bye-bye.